Welcome to Network Portal Tutorial 1, Exploring the Regulation of Genes. In this screencast, we will learn how we can use the Network Portal to explore the regulation of a gene of interest. Let's start with a search. In this example, we will explore the regulation of the HEM-A gene in Rhodobacter. HEM-A is one of two isozymes that catalyze the first step in tetrapyrrole biosynthesis. Previous studies show HEM-A is transcribed from two promoters and that the global regulatory proteins PER-A and FNRL regulate HEM-A transcription in response to changes in oxygen tension. Let's see if we can validate some of these results from our network model and then identify novel regulatory influences. After searching for HEM-A, we select the result from the organism of interest. The search results page gives an overview of the number of regulators for the gene, how many targets are regulated by the gene if it is a transcription factor, and which modules include the gene. We will go to the gene page. The Overview Gene page lists summary information, and more details can be accessed through the tabs. A module, or regulatory unit, refers to a set of genes that are co-regulated under a subset of conditions. As you will see in this example, looking at modules of co-regulated genes is helpful when analyzing gene function, and it is also useful in inferring regulatory influences. The Circular Regulon Explorer shows a high-level summary of the modules that include the HEM-A gene, shown in the context of the genomic location. The gene page also has a summary of gene information with links to outside web pages, an overview of the modules and regulators, and an overview of the discovered motifs. Functional annotations for the gene are shown from COG, GO, and KEG. The Network tab shows us the two modules of co-regulated genes that include HEM-A. We see all of the genes in those two modules, the four motifs discovered for those modules, and the regulators inferred to influence those modules. We can rearrange the appearance of this network diagram and click on Nodes for an information window. Now let's look at the regulators in more detail. Previous studies have identified a regulatory effect of PER-A on HEM-A, and we see that influence has been discovered by the network inference. We also see novel predictions. For example, RSP3238 and 2888 may also regulate HEM-A. These are hypotheses that could be tested experimentally. We also see potential motifs associated with these modules, which could be binding sites for these transcription factors. The Regulation tab shows a list of all of the influences on HEM-A that have been discovered by the inference algorithm. Interestingly, some of these regulators are also related to nitrogen metabolism. The Motifs tab shows more detail on the four motifs associated with modules containing HEM-A. We can further filter these motifs based on E-values to give us high-confidence motif candidates. The Functions tab shows functional annotations for the HEM-A gene from multiple ontologies, which is useful to connect regulatory influences with functional annotations. The Module Members tab shows the other genes that are members of the same modules as HEM-A. This helps to identify co-regulated processes. For example, here we see many photosynthesis genes. We can also use guilt by association to assign functions to hypothetical genes by using the functional enrichment information for the modules. Specific help information can be accessed on any page by using the orange help icon on the top of the page. You can also read the general help pages by using the Help tab. The Social tab lets you share your ideas or analysis notes with others in a collaborative discussion environment. To do further analysis of the regulation of this gene, we can use the FireGoose plugin. For example, we can broadcast the module member name list to other resources. In this tutorial, we have explored the regulation of the HEM-A gene. In addition to verifying the regulation of HEM-A by per-A, the regulatory network model identified other transcription factors that may regulate HEM-A and the motifs that may mediate that regulation. We made the insight that some of the putative regulators are involved in nitrogen regulation. We also observed an expected link between photosynthesis and porphyrin metabolism and identified functional associations for some hypothetical proteins. We learned how to use the network portal to explore gene regulation for a gene of interest, including discovering co-regulated genes, regulatory influences by transcription factors, potential regulatory motifs, and functional enrichments. We also saw how Network Portal can help you easily find related information from outside web pages through quick links and the FireGoose plugin. Enjoy exploring the networks, and please contact the Baliga Lab for more information.